Thank you. So in today's presentation, and I think it's a good segue to talk when we are talking about oncofetal ecosystem, oncofetal genes. I would like to bring you the concept of oncofetal ecosystem. And my lab focused mainly on oncofetal ecosystem, which we discovered together with peers a couple of years back now. And what does oncofetal means, which was nicely presented just now, is reactivation of fetal-like program in tumor cells. And it's a concept which was first proposed in 1960 in HCC. And ecosystem means basically an environment or geographical area. And what we are trying to understand is the reappearance of these fetal-like program in the tumor microenvironment. So as I just said, what happens is there are some genes, some cell types, some proteins, miRNAs, which are present in a fetal development, and they could reappear during regeneration and or cancer, right? And this is what we think is there are few hallmarks of oncofetal ecosystem. These are basically immunosuppression, proliferation, EMT stemness, and there is some kind of an underlying theme between embryogenesis, inflammation, regeneration, and tumorigenesis. And as I mentioned, the first report of oncofetal world goes back to 1960 in this seminal paper where they were trying to look into the similarity between fetal tissue and tumor. And one of the very famous example of oncofetal gene is AFP, which the name suggests, alpha-fetoprotein. But as we know, the oncofetal theme is not only limited to the proteins or antigen, but a lot of long non-coding RNAs such as H19 and microRNAs, which are also oncofetal. So I'm not going into those details, but what we are trying to understand is from the environment perspective. So all these studies were looking at oncofetal theme from a tumor cell perspective, for example, looking at these changes in the hepatocyte. And we asked the question into, I think, late 2018 or early 2019, what happened to the microenvironment? If there is a reprogramming happening in the hepatocyte, does it also influence the tumor microenvironment or not? So some of the aims we have is trying to look into the different components of the oncofetal ecosystem, signaling pathways between these oncofetal ecosystem, and if we can use this as a therapy response. So in today's talk, at the end of my talk, I'm going to a little bit discuss about how we think oncofetal ecosystem could serve as a potential biomarker of at least some of the therapy response. So I think as PS mentioned early on, the journey started with this paper where we're looking into our idea was to make a single cell atlas of HCC, but we did a little bit adventure and tried to include human fetal liver and some of the mouse models to see that what are the similarity between a developing tumor and fetal tissue. And to cut the long story short, we found some of the cell types which were shared between fetal tissue and tumor, and we identified some transcription factors which could be governing these phenotypes, and there some kind of a position where they were co-enriched in a tumor microenvironment. So this study was done using 14 HCC samples where we had adjacent normal and tumor. It's a multi-sectoral sequencing, which was very nicely described by Vivi in the beginning, so I'm not going into those details. But I think the uniqueness of this study was compared to most of the HCC atlas you will see is that we included 4 human fetal liver tissue, mostly coming from gestational age, 14 week, 16 week, 18 week, and 21 weeks, so second trimester, right? And what we did, we identified different cell types in the human fetal liver, human HCC, and we could also identify which cell types were tumor specific, which cell types are present in the core and periphery. And then we use this information to look into the tumor specific cell types, and then we ask the question which of these are also present in the fetal liver. And in the original study, we identified 2 major cell types which were shared between fetal tissue and tumor, and these were macrophages. There were 3 different macrophage populations, and we found one of the macrophage population, which we call TAM1, was more fetal-like. And among 5 different endothelial populations, we find a endothelial population of PLBAP, which is more Onco-Fetal. So since this study is published and almost now 1.5 years ago, I'm not going into detail of this paper, but this was a summary figure, and we found that this program is driven by proliferating hepatocyte, which reprogram endothelial cells and then lead to the reprogramming of the macrophages and T cell. And I was very excited early by looking at Arjun's talk, where she's showing that how this epigenetic rewiring could be leading to these oncofetal cells. And I think we did not look into the epigenetic profiling or epigenetic aspect in our study, but it will be very interesting to see these VEGF-alpha positive hepatocyte, if this epigenetic reprogramming is happening specifically in these cells, and what role possibly could be led by some of the signaling pathways such as VEGF-alpha. But when we were finalizing the study and manuscript was almost in the final round of review, this very nice paper came in NEJM, which first time suggests that an immunotherapy, which is combination of VEGF inhibitor and PD-L1 inhibitor, is actually working in the clinic. And then in last year, the study was, we have a more advanced data in the study, which shows that at least there is 13-month survival benefit with this immunotherapy compared to the sorafenib. So since then, we started asking questions that we know there's an immune cell which is oncofetal-like, and we know there's endothelial cell, stromal cell, which is oncofetal-like. Are there other cells which could be oncofetal in the tumor microenvironment? And we found one more population which is stromal, but since the work is currently in review, I can't reveal the specific marker of this population, but then we found that there are more oncofetal cells in the tumor microenvironment than were initially discovered in the first study. And found that there's particularly one cluster which is present in both fetal liver and the tumor and absent in the healthy tissue as well as normal tissue. So in a previous study, we did not have healthy tissue, but now in this study, we have downloaded some healthy human liver tissue, and we can compare that if there's a differences between healthy tissue versus adjacent normal or not. So what we did next is that we, once various data was published, and Shinchi and Pierce were very generous in sharing this data with us. So we combine our single cell RNA-seq data with this bulk RNA-seq, which is Planet, and we tried to use some of the algorithms such as CyberSortX, and we now try to deconvolute this bulk RNA-seq data to see which cell types are present. So as you know, we cannot do single cell in a lot of patients, and single cell is more of a prospective study. So we try to deconvolute this bulk RNA-seq data, which has more mature cohort, and we know what happened to the patient in terms of response. So idea was to look into this heterogeneity of HCC, and as it was discussed earlier, that HCC is not only a heterogeneous disease in terms of the genotype and phenotype, but kind of stages which appears in the clinic and therapy given to them. So most of this patient in this cohort are coming from the very early stage, and these are surgical resections, right? So we wanted to understand what happened to these patients when we collected the sample, we profiled them, and these patients were given surgery as a treatment. So what happened to the patient as surgery, in how many patients surgery worked or like relapsed happened early versus late. So this is the compiling lot of data in just one single slide. And as I said, this work is currently under review, so I can't reveal much, but what we identify, some of these fetal-like cells could really classify these patients who will have more chance of relapse compared to another group of patients. So found these two group of relapsed patients, one group with very high oncofetal cells. Next, what we did, we look into the spatial transcriptomics. This is, we are looking at nanostring, whole transcriptomic atlas. We're looking at 18,000 genes, but we are looking at two patient groups. So one patient which did not relapse and another patient who relapsed very early, so within 6 months, right? And actually, this patient relapsed within 3 months. So we did this spatial transcriptomics, and now we looked into the areas based on the immune, stroma, and tumor markers. And we also did same experiment on the fetal liver, and what we could identify, the patient which recurred earlier had more similarity in terms of the transcriptomic profile compared to the fetal liver tissue. And again, when we look into these two patients, we found that patient who recur early has more oncofetal cells compared to the another group of the patient. And finally, we use this very new technology, which is still not commercial yet, but basically it's allow you to look into the subcellular resolution at the spatial transcriptomics. So this is BGI stereo-seq or STomics, which basically allow you to look into the nanometer resolution. And in this case, we can just go back into the single cell resolution. As we know, most of the spatial transcriptomics methods does not give you single cell resolution, right? So we found that this not only give you single cell resolution, but we can get a lot of good number of detected genes here. And this is some of the QC analysis. But what we did here, we could look into different oncofetal cells, and by doing this neighborhood analysis, we can identify where in the tumor these oncofetal cells, which is or macrophages, endothelial cells, and this new stromal population co-localize, and what is the property of the tumor cell in this area. And finally, this is what we are developing now is a multiplex panel where we could look into different cell population as well as the immunosuppressive marker, and we can look into what are the property of the tumor in the oncofetal high versus low area. So finally, once we know that oncofetal ecosystem has some implication in predicting response after surgery, now with in the collaboration with peers and other clinical collaborators in Australia, we have launched this study where we're looking into the advanced HCC via biopsying these patients in Perth, and we're looking into if we can predict the response to the immunotherapy in these advanced stage HCC, which was recently funded by NHMRC last year. So in summary, what I'm trying to convey is that oncofetal reprogramming is associated with immunosuppressive microenvironment, which we discovered early on. But I think the most exciting thing now is that it's not only basic biology discovery, but we can use it for predicting therapy response. And I just give you an example in the surgery, but I think it has implication beyond surgery. And the single cell and imaging based methods allow you to predict that both VEGF and PD-L1 are enriched in oncofetal ecozones, suggesting that these immunotherapy response could be predicted by presence and absence of the oncofetal cells. My original oncofetal discovery was in collaboration with PS, and this new work is also in collaboration with PS and Florent, and Z from Shanghai is leading this thing, and Ria Pai from my lab, who's my PhD student, has done a lot of spatial transcriptomics and imaging, and collaboration with BGI and NanoString for spatial transcriptomics work I just showed you. So thanks for listening to me, and I'll take any questions. Thank you.